Good afternoon. We have spent the last 10 sessions really focused on what viruses are doing, how they're built, how their genomes are replicated, how they're assembled. Today I want to focus on the effects on the cell, and that's why we call this the infected cell. We have talked a lot about how virus replication depends on many aspects of host machinery, of course, like metabolic machinery, biosynthetic machinery, like transcription, protein synthesis, signal transduction pathways, trafficking systems, moving things from place to place. So what I want to do today is give you a picture of how virus infections impact these processes. Not only is virus reproduction dependent on them, but often infection modulates them to direct them towards virus replication. And this is a relatively new area of investigation. We're just beginning to learn a lot about how during the reproductive cycle the cell is really changed. And sometimes the changes are subtle and the infected cell can live for a long period of time. But other times they are very dramatic and they lead to cell death. We're going to talk about 4 different areas today. Signal transduction, what viruses do to alter that. Gene expression, like transcription, the production of mRNA, protein synthesis, metabolism. We've learned in the last 10 years or so that viruses actually turn up the thermostat. They make the cells increase their metabolic activities. And finally, we'll revisit something we've mentioned a couple times, the remodeling of cellular organelles. This happens throughout the virus-infected cell, but we'll focus on a couple of examples in the cell cytoplasm. So let's start with signal transduction. This is probably the bane of every student's existence when an instructor throws up a signal transduction pathway with zillions of arrows going in all different directions. And you know, some people would like you to memorize these. Fortunately for you, I do not. Don't have to memorize this. You don't even have to write this on your 5x7 card. I would never ask you to remember any of these details. What I want is you to understand the overarching principles. Signal transduction pathways are the way the cells sense what's going on around them, either chemicals or touching another cell, and respond. So from the outside to the in, even within a cell, signal transduction pathways govern just about everything about how the cell works. And virus infections impact these, for sure, and we're beginning to learn more and more. I'm going to give you a couple of examples of how that happens today. And here on the left is a very important signal transduction pathway. It's called the PI3K-AKT-mTOR pathway, and that name is why many people hate these. Horrible name, but it's based on some of the main players, PI3K, AKT, and mTOR. But this one is one of several important for translating signals from the cell surface to some effector. So let's say, for example, if there are growth factors in the medium, and these, of course, stimulate cells to divide. They bind receptors. Receptors here, that little purple one, RPTK, receptor tyrosine kinase. The ligands bind, the growth factors bind, and then they initiate a signaling pathway, which involves phosphorylation and other modifications of the intermediates. And eventually, that would lead to increased mRNA translation, cell growth, and so forth. So that's how the signal is transmitted from the cell membrane all the way into the cell actually doing something. And these pathways are full of arrows because many things can happen. Often multiple pathways converge on each other, and a signal at the surface can have several different outcomes depending on other factors as well. So it's necessarily complicated because cell life in the cell is complicated as well. Now, when I show you these diagrams, there'll be a few of them. The green arrows, of course, mean stimulation. So something is stimulating something else, and the red arrows mean inhibiting. And so, for example, if a growth factor binds its receptor on the cell surface, that will lead to stimulation of PI3 kinase activity, possibly in the form of phosphorylation. Now, on this slide also are written names of different viruses and how we've learned they impact this particular pathway. And you can see some viruses stimulate particular parts of this pathway by the green arrows, and some viruses are inhibiting parts of the pathway. And this depends on what's supposed to happen. For example, viruses stimulating this pathway could lead to an increase in protein synthesis. Viruses inhibiting the pathway could lead to preventing apoptosis, programmed cell death. So it's different for each virus, but we're learning, as you can see here, that viruses are impacting many different parts of this pathway. So let's take a look at some examples of signaling and how it's modified by virus infection. This is a really interesting one. Virus binding to receptors actually initiates signaling pathways that facilitate virus uptake into cells. This makes perfect sense, but we didn't know this for many years until we had the tools to sort out these signaling pathways. Here are 3 examples, 3 different viruses. On the left is adenovirus. Binding to its receptor on the cell surface initiates a series of signal transduction pathways. The details aren't important, but what's important is the outcome. And you can see some of the players on the right in this pathway. But the outcome is that the actin filaments which are existing just below the plasma membrane, they have to be rearranged. So endosomes moving into the cell, which are going to take up adenovirus, they have to move through the actin filaments, and that's disassembled essentially by the signaling that's induced by virus binding to its cell receptor. So basically the virus is tapping into pathways that already exist, because in order for vesicles to come in from the surface, you have to disrupt this actin filament structure, and so the virus does that, and then it can move into endosomes. In the middle, similar situation, we have influenza virus binding its receptor. In this case, it is actually a separate effect of binding. The virus binds near what we call lipid rafts. These are specialized microdomains in the plasma membrane, and the virus binding causes clustering of receptor protein tyrosine kinases, these RPTK molecules, and that initiates a signaling pathway that also loosens the actin filament structure so the virus can enter by endocytosis, but also activates a vacuolar proton pump in the endosome membrane. That's that little yellow cylinder there, and you can see a green arrow coming from these particular signaling pathways. So this same signaling that allows the endosome to move through the cell increases the activity of this proton pump, pumps protons into the endosome. Of course, that lowers the pH, and that's what uncoats the virus. That's what leads to HA-mediated fusion. So the virus is triggering events that are needed for efficient entry, both moving through the endocytic pathway and low pH. And finally, on the right is Ebola virus. Again, the virus binds to cell receptors taken up by macropinocytosis, this non-receptor-mediated endocytosis pathway. It's a separate pathway. And this virus gets into an endosome. But early on, the binding to the cell surface, again, stimulates PI3K-AKT pathway to allow the virus to remain in an endosome. So normally, without this stimulation, the endosome would move to a non-productive part of the endosome pathway. The virus wouldn't be able to fuse and get out into the cytoplasm. So the virus binding to the surface stimulates this pathway, which inhibits movement of the endosome to a non-productive pathway. It's what we need to know about that if without this stimulation, the endosomal pathway would continue and the virus wouldn't be able to get out. So the inhibition of that trafficking keeps the virus in an endosome from where it can then fuse and get out into the cytosol. So 3 examples of how virus infection modulates signaling to facilitate virus entry. Sometimes different viruses affect the same pathway, as you might guess. You saw some examples of that in the previous slide. Here, a couple of other examples of, again, the PI3K, AKT, mTOR pathway. And here we have some viral proteins on the left interacting with a subunit of PI3K. You have a hepatitis C virus protein, you have a rotavirus protein, you have an adenovirus protein. They're all binding PI3K, and they're stimulating its activity, stimulating signaling through AKT, and this results in cell survival, You have inhibition of programmed cell death, apoptosis. Now that makes perfect sense. The viruses need the cell to be alive in order to replicate. Translation is also stimulated, so viral mRNAs can be efficiently translated. Now hepatitis B virus on the right also one of its proteins also interacts with a subunit of PI3 kinase, again stimulating cell division, inhibiting apoptosis, and stimulating translation. And if it were not for that, the Hep B genome's transcription would be inhibited. So the effects of the Hep B protein on this pathway alleviates that inhibition. You can see here is another virus protein, Synbis virus, and alpha virus is also interacting in this pathway, leading to cell proliferation, increased translation, as is a protein of a virus called human herpes virus 8. And this is actually a viral protein that forms a multipass plasma membrane channel, and it interacts with PI3K subunit also to stimulate beneficial functions in the cell. So many diverse viruses can intersect the same pathway to have common effects. We also have an interesting example of how, so it doesn't have to be a viral protein with activity that affects these signaling pathways. Here's an example of a viral RNA that can block AKT activation to induce apoptosis. So far we've been talking about turning up AKT activation. Here's a flavivirus. This particular observation holds for a number of different flaviviruses. Flaviviruses include West Nile and Zika virus, dengue virus. Here's a flavivirus particle. We have looked at those before. They're enveloped with the glycoproteins flat on the surface. It has a plus strand RNA genome that's translated into a single long polyprotein. When this RNA gets into cells, one of the responses of the cells do not like foreign nucleic acids coming into them. They have lots of defense mechanisms to deal with that. And one of them is that enzymes are present that will degrade viral RNAs. There's a whole host of degradative enzymes. One of them is shown here, XRN1, which is a 5' exonuclease. It chews in from the 5' end. Now you see there at the 5' end of this message there's a phosphate. There are also enzymes in the cell that first take off the cap structure. They're called decapping enzymes. And that sets up the RNA for XRN1 to start chewing it down. So this is a cell defense mechanism. And viruses have ways to counter this. We'll see. But in this case, the digestion by XRN1 produces a product that blocks AKT activation. So we have XRN1 chewing down through the 5' non-coding region, through the whole open reading frame, which is most of the RNA. It's not really drawn to scale. And then it can't go any further than the 3' UTR. 3' UTR, which is relatively short compared to the rest of the RNA, is highly structured, and it's too structured for XRN to chew through. So what's left is a short, highly structured RNA. It's called a subgenomic flaviviral RNA. And that RNA binds AKT, and it blocks its activation. You can mutate the genome to prevent the generation of this small RNA. Okay, you can make a small mutation in the 3' end, so XRN no longer will make this. Those viruses replicate fairly well in cell culture, but they don't form plaques. So here's a plaque assay with wild-type virus, and on the right is a mutant where this subgenomic RNA can't be made. It actually does replicate well, but it doesn't form plaques. And if you inject this virus into mice, it doesn't cause disease. So this small RNA is absolutely essential for killing cells and causing disease in mice. And what it does is it promotes programmed cell death. Programmed cell death, apoptosis, some viruses inhibit it, some promote it. It really depends on how long the replication cycle is, if cell death is needed to let the viruses get out of the cells. You can't make any generalizations. In this case, this RNA binds AKT, it inactivates it, and that in turn reduces the concentration of a protein called BCL2, which is an anti-apoptotic protein. The detail's not important. What is important here is that this small RNA promotes apoptosis so the cells can be killed and it can spread within an animal. So I think that's a really interesting example of a non-protein mediated modulation of signaling pathways. Now moving into gene expression. Viruses impact almost every phase of cellular gene expression, starting with transcription. So here we have a picture that you've seen before, which is the pathway of transcription and RNA processing, starting from a double-stranded DNA in the nucleus. The gene is transcribed from a promoter. Remember, it's transcribed, polyadenylated, spliced, capped, and then exported into the cytoplasm. And these red letters here show you where different viruses interrupt this process. At every stage, they make gene products that interfere. For example, these viruses here, polio, VSV, and an alpha virus. An alpha virus would be SynBis. We mention those now and then. They'll inhibit transcription or capping. And they do so by inhibiting the enzymes or the factors that are needed for it. For example, poliovirus encodes a protease that cleaves transcription factors of the cell to inhibit Pol1, Pol2, and Pol3. I'm showing here just Pol2, but polio cleavage is actually the Tata box binding protein that cleaves required for all three. Inhibits RNA synthesis pretty much globally in the cell. You may say, why? The virus is an RNA virus. Why does it matter? So it doesn't have any competition for the RNA triphosphates. And then in terms of mRNAs, there's no competition for the translational apparatus. When everything devoted towards viral reproduction. And also capping can be inhibited. Splicing and polyadenylation can also be inhibited. As another enterovirus, influenza A virus, a herpes simplex virus protein inhibits splicing. Enterovirus also inhibiting polyadenylation. And then, of course, we have to export the mRNA. And there are virus proteins that interfere with export. Remember, export has to occur through nuclear pores. And those are listed here. Polio, VSV, adenovirus. There are some viral proteins that will degrade mRNAs. That's shown here. These are encoding proteins that chop up the mRNA. And a really interesting question is, how do they avoid chopping up their own mRNAs? We don't have good answers for that yet. And finally, these mRNAs, of course, have to be translated. And many, many viruses inhibit cellular translation so that only their mRNAs are translated. So they have the whole translational apparatus devoted to the synthesis of viral proteins. So you can see this is a really global alteration of gene expression. You take an uninfected cell, the virus infects it, and sometimes within an hour or two, all these major biosynthetic processes are inhibited. And that's in part why cells die. We also have apoptosis and other kinds of programmed cell death, but often this cessation of biosynthetic activity simply kills the cell. Let's look at some of these in some detail. And remember, the idea of doing all this is to reduce competition of cellular mRNAs with viral RNAs for the translational machinery. This is also a picture I showed you earlier without the viral proteins on it. This is the process by which a pre-mRNA is spliced and exported from the nucleus. Now remember, splicing occurs on the spliceosome, and this is a big protein RNA complex. And during splicing, small proteins, shown here, like CBC, T-Rex, isn't that a great name for a protein? T-Rex, SR, etc., are all deposited on the RNA, and then they mark the RNA for export by the nuclear export machinery. And so here we have a number of viruses which are interfering with different aspects. Here we have this herpes virus protein, which binds at the SR protein and prevents splicing from happening. So cell mRNAs can't be spliced. Now, if you are wondering, why would that happen in a virus-infected cell if the viral mRNAs have to be spliced? Well, the answer is most of the herpes virus mRNAs aren't spliced. They don't have introns. So that's a good evolutionary strategy. Then we have some viruses here. Influenza virus, the protein called NS1, can interfere with polyadenylation. It binds to specific proteins needed for polyadenylation at the 3' end, and the flu protein can interfere with that. And then the rest interfere with export. We have the poliovirus protease cleaving NUPs. NUPs are nuclear export proteins that are needed to bring material out of the nucleus. It's cleaving them, so export ceases. And you may say, well, why would a virus muck up the export machinery? Because it doesn't care It doesn't have anything to get out of the nucleus. It replicates entirely in the cytoplasm, yet it prevents cell mRNAs from getting out and competing. So polio inhibits export by cleaving NUPs. Adenovirus binds to members of the export protein family. Some two viral proteins here prevent export. We have VSV, M protein binding, NUPs as well. So you can see just many ways to mess up this mRNA synthesis, processing, and export pathway by viruses. Now let's talk a little bit about another way that virus infections shut down host processes, and that's by degrading cellular RNA. We've mentioned this already, but I just wanted to bring it up again in some detail. So as you know, in cells, mRNAs have half-lives. They don't all exist for the same amount of time. They could be less than 30 minutes. From their genesis in the nucleus, they could go in the cytoplasm and exist for less than 30 minutes. Sometimes they exist for more than 12 hours. It depends on how important the protein is and how much you need of it. And we have ways to regulate the half-life of messages. There are specific signals in the messages, and we have a bunch of enzymes, which I've already introduced, that degrade messages when we don't need them any longer. And here's an example of some mRNA turnover systems. These divided into two categories, either 5 to 3' decay or 3 to 5' decay. These both start with the recognition of the poly A tail on an mRNA by this protein here, blue one, and it's chewing away the poly A tail, and for some reason that signals then the rest of the decay to occur. On the left, the 5 to 3' occurs. First, the cap is removed by decapping enzyme. That's a DCP, decapping protein. And then once the cap is removed, here's XRN coming in and chewing in from the 5' end. That'll just degrade all the mRNA. But again, you have to first remove the poly A tail, and then the cap, and then XRN comes in. Or for 3 to 5' decay, removing the poly A tail signals for a degradative structure called the exosome to come in, and then another protein chews up the rest of the RNA. These are not only cell mechanisms for regulating their own mRNAs, but they are used to degrade viral mRNAs when they're produced in cells. So, as you might guess, viruses encode antagonists. And here's one example in cells infected with poliovirus. These nucleases, PAN, DCP, and XRN1 are all degraded in poliovirus infected cells, so to allow the viral RNAs to exist without being degraded. The other side of the coin is some viruses encode proteins that chew up the host mRNA. So the host has its own set, and then the virus makes its own as well. So here is an example of several viral proteins. So here on the left is a cellular mRNA in the process of being translated, although if it were viral, it would look exactly the same. You have a cap at the 5' end, and then a ribosome is translating, and there's your poly A tail. Translation occurs on circularized mRNAs, so the 5' and the 3' ends are brought together, and we think that allows the ribosomes to go around them many, many times. SARS coronavirus encodes a protein called NSP1, which seems to recognize the ribosome and then starts to chew up mRNA. You can see that red protein there chopping it up. And then once an mRNA is broken, that's a signal for XRN1 to come in and destroy it. So the viral proteins are kind of seeding the destruction of the cellular mRNAs by the cellular exonuclease XRN1. So that's one example, SARS coronavirus protein. Herpes simplex and HHV8, human herpes 8, both encode proteins that recognize the cap structure. They also will start degrading the RNA. VHS and SOX are the proteins, and that again leads to XRN1 degradation. And then vaccinia virus encodes actually a decapping protein. Two decapping proteins, removes the cap, which of course is a sign that XRN1 can come in and degrade that mRNA. So you see the viral proteins don't do all of the degradation themselves. They just mess up the mRNA enough, they either nick it or take the cap off, and then XRN1 will come in and chew it up. And again, the question is how virus and host cell messages are distinguished. We don't really know the answer, although some viruses don't have caps, and so they escape the cap-dependent interaction. One more example of an RNA that can modulate this cellular response. And this is an example from hepatitis C virus. And hepatitis C virus we'll revisit in a bit. As the name implies, causes hepatitis, inflammation of the liver. It's a flavivirus. It's an enveloped RNA virus. And this virus is hepatotropic. It is liver specific because of a small microRNA that is produced only in the liver. It's called miRNA or miR122. As you know, microRNAs are produced in our cells by transcription of genes and processing of those transcripts to short 21 nucleotide RNAs, which will then silence gene expression either at the level of causing mRNA degradation or translational inhibition. This particular one, which has roles in cholesterol regulation in the liver. So the microRNAs that we make are for our purposes pretty much, but this one happens to be involved in allowing hep C to replicate in the liver. And what this microRNA does, it binds to the very 5' non-coding region of hep C RNA. So here's a schematic of the genome. The 5' non-coding region is in green. It's highly structured. And then the coding region is downstream from it. And the very 5' stem loop is expanded below. And you can see there are two copies of miR122, one at the very 5' end and the second one downstream a bit, which are base pairing with the RNA. And they block degradation by XRN1. If not for these miRs, the RNA would be degraded by XRN1. And in fact, that's what happens in other cells when hep C gets into a kidney cell or intestinal cell. There's no miR122 because this is a liver-specific microRNA, and the virus won't replicate in those because the RNA is degraded. So it's another example of how a virus can get around cellular defenses, an XRN1 based defense here by utilizing a cellular RNA. So it's evolved to be able to do that. We'll see later when we talk about antivirals that targeting miR122 has therapeutic value. Drugs have been made which basically bind miR122 in your liver and sequester that microRNA and prevent it from binding to the hep C genome. So the virus will not replicate in your liver any longer. It will be degraded by XRN1. Our first question is, which of the following is a consequence of viral proteins modifying signal transduction pathways to promote viral replication? A, polio inhibition of transcription by RNA Pol2. B, herpes simplex virus protein blocking pre-mRNA splicing. C, disruption of actin filaments to allow endocytosis. D, initiation of mRNA degradation by viral proteins. Well, 67%, most of you got C, disruption of actin filaments to allow endocytosis. So that's the signaling pathway involved in promoting viral replication. All the other ones certainly occur, but they're not involving signaling. I want to talk a little bit about translational regulation because this is a big point of intervention from the virus and the host side. So remember, all of our 7 viral genome classes all have to get to mRNA, which can be translated by the host cell because viruses don't encode the translation system. And every virus figures out some way to inhibit host translation by many different mechanisms. Again, so that the viral mRNAs are preferentially translated. That makes perfect sense. So let's look at some examples of this. First, to make sure that you understand mRNA translation, mRNAs, of course, have a cap at the 5' end, an AUG or initiation codon, and they're polyadenylated. And the cap is recognized by the initiation complex. There's a protein called EIF4E that binds it. And then via a series of protein-protein interactions, EIF4E binds EIF4G, which binds EIF3, and that recruits the 40S subunit of the ribosome. And that pre-initiation complex slides down to the mRNA, finds the AUG codon, and then at that point the large subunit comes in, and now we are ready to make protein. And this initiation complex has with it, besides all these proteins, the initiator tRNA MET. And that's going to be what's first put down at the AUG codon. So this is called 5' end dependent initiation because you need to have a free 5' end with a cap. Now is an example of viral inhibition of protein synthesis. So on the left is a graph rate of protein synthesis on the y-axis and time post-infection on the x-axis. These are cultured cells that have been infected with poliovirus. And you can see by 2 hours post-infection, the rate of protein synthesis has gone down substantially, whereas uninfected cells, it would remain the same. And then starting at about 3 hours, the rate goes back up. And of course, that's the viral mRNA being translated. And if you look on the right, this is a protein gel where we've taken extracts of the cells at different time points and run them out on a protein gel. And they're labeled with S35 methionine. So you can see at time 0, all these bands are cellular proteins. There are lots of cell proteins, so they make up a big smear. But then you can see that the cell protein synthesis eventually goes down, and it's replaced by viral proteins, which are the letters shown on the right there. So this is just one example of many of how a virus interferes with cell protein synthesis. This happens to be really quick, 2 hours in these particular cells. Some viruses take longer. But again, the idea is to have only viral mRNAs being translated by ribosomes. There are many mechanisms by which this happens. And here are some of the mechanisms that are involved in modulating the cap recognition step of protein synthesis. Remember I said EIF4E recognizes the cap on the mRNA and it brings the ribosome to it. So here are a couple of mechanisms for a few different viruses. On the top, poliovirus and other picornaviruses encode proteases that cleave this red protein, EIF4G. EIF4G is the bridge between the cap binding protein and eventually the ribosome. Another protein would be in here. Here it's shown at the bottom. You see EIF4E binding the cap. It binds EIF4G, EIF3, and then the ribosome. And so polio and foot and mouth cleave EIF4G. It separates the two parts of EIF4G. So no longer will EIF4E be able to bring the ribosome to the mRNA because the EIF4G is broken. So that is why cell protein synthesis was inhibited in that previous slide, because of cleavage of EIF4G. A couple of other mechanisms are shown here. Some viruses induce dephosphorylation of EIF4E. The binding of EIF4E to EIF-G is dependent on phosphorylation of EIF4E. So if you dephosphorylate, 4E will not bind 4G. So this makes sense if you need an intact EIF4G, if the viral mRNAs require that. And then we have a third mechanism here, which involves a second protein that is involved in regulating the activity of EIF4E. It's called 4E-BP1, 4E binding protein 1. And when it is dephosphorylated, it binds to 4E and it prevents 4E from binding EIF4G. So some virus infections result in dephosphorylation of 4E-BP1 so that they will sequester 4E away from EIF4G. So these are just a couple of many, many mechanisms we've learned about how viruses induce shutoff of host cell translation. Now you may be wondering why polio and other viruses, why would they induce cleavage of EIF4G if that's an integral part of the translation initiation pathway? Well, it's because those virus mRNAs do not require a cap. They don't have a cap. All the picornaviruses do not have a cap. Hepatitis C virus, there's no cap. So they don't care about the cap binding protein. Now at the top is the way cell messages are translated. Again, 4E binds the cap and brings in the ribosome via that interaction. Coronaviruses, they don't care if 4G is cleaved because they don't have caps to recruit 4E anyway. But 4G is involved in the translation of their mRNAs. It binds actually directly to the highly structured RNA at the 5' end of the genome. And it can bring in the 40S subunit that way. And hepatitis C virus also. In fact, here the EIF3, it doesn't even need EIF4G. EIF3 itself can bind to the viral RNA and recruit the 40S ribosome. So this is why you can cleave EIF4G in cells infected with these viruses. You do not affect viral protein synthesis because it is not 5' end dependent. So what is it? Well, it's internal initiation because the 4G recognizes, or EIF3 recognizes the RNA internally and the ribosome binds internally. This was first discovered in the genome of poliovirus when it was first sequenced. It was a very long 5' non-coding region found before the first AUG that begins the polyprotein. And this sequence is highly structured, I'll show it here, and binds, in the case of polio, EIF4G. So here are 5' non-coding regions for a number of different viruses that do not require a cap to initiate translation. So here in the upper left, we have the poliovirus 5' non-coding region. We have encephalomyocarditis virus, hepatitis C virus, iris, and a couple of other viruses as well. There's an insect picornavirus, but you can see they're all highly structured. And in the case of polio and encephalomyocarditis, 2 picornas, the EIFG can bind directly to this structured RNA. So it does not require a cap, it doesn't require EIF4E at all because there's no cap, it does not require the N-terminus of EIF4G. So that's why that particular mode of shutoff works by cleaving EIF4G. Other viruses do it in other ways because not all viral mRNAs are uncapped. Now, when viruses infect a cell, there's a lot of responses, as I've already told you, one of them being degradation of viral mRNAs, but another is that the cell attempts to shut down protein synthesis very early in infection. The idea being that that's not good for the cell, but you could stop virus infection early on, you would prevent spread to other cells within the organism. And of course, as you might gather, viruses counter that. So I want to tell you that story here. And to do that, we need to look at the initiation step of translation. So again, we have our mRNA here in the upper left with a cap. RNA is in green. And the initiation complex has bound to it. And that initiation complex, again, contains a tRNA met. That's the initiator tRNA. When the AUG codon is reached, the large subunit comes in. In fact, there's a molecule of GTP bound to this tRNA, the initiator tRNA. It's hydrolyzed to GDP, and then it has to be recycled in order to do another round of initiation. Notice that GTP is bound to a protein called EIF2, which in turn binds the MET tRNA. And then when the large subunit comes in, GTP is hydrolyzed, we now have GDP EIF2. The recycling is done by a protein called EIF2B. This binds GDP and EIF2, and it exchanges the GDP for a molecule of GTP. So it's a guanine exchange factor. There are lots of these in cells. So now you have EIF2 with GTP, and then that will bind another MET tRNA, and that can then participate in another round of initiation. So this is essential for initiation, having GTP bound to EIF2. In our cells, there are a number of different protein kinases that will phosphorylate a subunit of EIF2 and prevent this recycling reaction. And here's the phosphorylation of EIF2 shown at the bottom. So we have GDP EIF2. This would then bind the EIF2B protein in order for the GDP to be exchanged, but the problem is GDP can't get out of this complex. You end up sequestering all the free EIF2B in the cell, because again, the phosphorylation of EIF2 makes this complex bind so tightly to EIF2B that it's never recycled. So you bring down levels of EIF2B, you can't recycle GDP anymore, and you inhibit translation. So that's the cell's response to a virus infection. And the way the cell does this, it can sense virus infection. I'll tell you how in a moment. And these protein kinases are activated. They're called EIF2 alpha kinases. That's a subunit of EIF2 that gets phosphorylated. Here are 3 different protein kinases that can phosphorylate EIF2 alpha. And 2 of them are relevant to virus infection. One of them is called GCN2, which phosphorylates EIF2 under conditions of starvation, amino acid starvation. So if you starve cells or even animals, they have no amino acids to make proteins, so this protein kinase shuts off translation. There's no point in trying to translate if you have no amino acids. PERK and PKR are 2 EIF2 kinases that are active in virus infection. PERK responds to ER stress, endoplasmic reticulum stress. When viruses, especially viruses with glycoproteins, replicate, they make lots of proteins in the ER, and that stresses the ER. And there are proteins in the membrane, you're going to see a picture of one shortly, that can sense that stress and phosphorylate EIF2 alpha. The point is, no point making more proteins until you can get rid of that stress. And there's another one called PKR that's activated by double-stranded RNA. So again, these are sensors of virus replication that can sense when a virus is replicating and shuts down translation by virtue of phosphorylating EIF2. All right, so if you've got that, let's move on to how this PKR works. PKR is made in the cell in an inactive form. This is a cartoon of PKR. There are two double-stranded RNA binding domains in brown and orange, and then there is a kinase domain, the KD. As this protein is made in a cell, it's inactive. It's unable to phosphorylate anything. In a virus-infected cell, however, there is often double-stranded RNA present. That's shown there in the middle of the slide. We don't usually have double-stranded RNA in our cells unless there's a virus infection. For example, RNA viruses, of course, make double-stranded RNA intermediates when they replicate. And DNA viruses often do transcription from both strands, so you'd have the opportunity to have double-stranded RNAs made. Those double-stranded RNAs bind PKR. You can see the second panel. These double-stranded RNA binding domains mediate the binding of PKR to the double-stranded RNA, and then adjacent PKRs can phosphorylate each other, and then they become active. They can then go off and phosphorylate EIF2 alpha. So this protein kinase in your cells is normally not active. You don't need it except under certain conditions. And one of those conditions is a virus infection, which is signaled by the production of double-stranded RNA. Now at the top is another pathway of activating PKR. There's a protein called PACT, which can actually bind PKR and activate it. So in our uninfected cells, there's some conditions where we don't want protein synthesis to occur, and that's what PACT is for. PKR, again, to review, as it's translated and produced in a cell, it's inactive, but actually, we'll see later, it is induced by virus infection. It's actually induced by interferon. It's activated by sensing double-stranded RNA, leads to inhibition of host translation, and that is a signal for programmed cell death. So actually, phosphorylation of EIF2 alpha triggers programmed cell death. The idea is the cell dies before the virus can spread. Every virus on the planet has at least one way of evading PKR or a similar kinase, because this is so potent. If every viral genome did not encode an antagonist of this pathway, there would be no viruses around. It's really that good. But there has been a continual arms race over the millennia between viruses and cells. You know, viruses antagonize PKR, the cells change to fight back and forth, back and forth. So here is one example of a viral gene product that antagonizes PKR, and it's a small RNA produced by the adenovirus genome called VA RNA I. And what VA RNA I does, it's shown here on the left, VA RNA I will bind one molecule of PKR, and only one. You can see that on the lower left. Not two, and of course two PKRs need to bind next to each other in order to activate each other, so VA RNA essentially prevents activation of PKR. So you get no phosphorylation of EIF2 and protein synthesis continues. So again, this is a viral antagonist of PKR. And just to remind you, if there were no VA RNA present, the virus would be making double-stranded RNA, which would activate PKR, which would eventually shut off protein synthesis. As I said, every virus that we have studied has some antagonist of this pathway. It's that powerful. Here are some of them shown here. And the pathway is interfered with at multiple places by different viruses. Some viruses interfere with EIF2 itself. They make what we call pseudo-substrates. It looks like EIF2, and it's phosphorylated by PKR, but PKR ignores the EIF2 in a cell because there's so much viral protein present that you don't get shut off of host translation. Some viruses encode inhibitors of the kinases. So here's PERK, by the way, which is a kinase, which is actually a transmembrane protein in the endoplasmic reticulum. The blue part senses protein misfolding in the ER and will become activated. It dimerizes the two subunits phosphorylate each other. Well, viral proteins can inhibit them. And then there are a whole series of viral proteins that interfere with PKR itself. You've heard about one of them, Adenovirus VA RNA, which is an antagonist of RNA. Basically, it prevents RNA activation of PKR, but they're also double-stranded RNA binding proteins. They bind up the double-stranded RNA, so PKR could not. And they're also antagonists of the protein itself. So this is just a partial list. I just want you to appreciate this amazing fact that every virus has to antagonize this pathway, otherwise it would be out of existence. It's really a very powerful pathway. Our next question is, PKR is an interferon-induced enzyme that is activated by blank, leading to phosphorylation of blank and inhibition of translation. So we have, to fill in the blanks, GDP, EIF2 alpha, double-stranded RNA, EIF2 alpha, double-stranded RNA, EIF2B, single-stranded RNA, EIF2 alpha, or maybe none of these is right. So what activates PKR? Most of you got double-stranded RNA, that's right, and it phosphorylates EIF2 alpha. Some of you picked double-stranded RNA and EIF2B, but EIF2B is not phosphorylated, and it is not single-stranded RNA. So we talked about how EIF2 alpha gets phosphorylated, shuts down translation globally. Another response is the formation of stress granules. These are shown here. When you stress a cell by virus infection or withdrawing amino acids, again, there's no point in making protein under these conditions. So mRNAs that are in the process of being translated on the upper left, they are shunted into these cytoplasmic structures called stress granules. They're made up of a variety of cell proteins, and some of them are shown here, G3BP, TIA1, TIAR, and viral mRNAs. They're very discrete structures in the cytoplasm. In the air, the mRNA is silenced. It's not translated. So the idea is, for the cell, if there's a stress condition, put your mRNAs in a stress granule, and then when the stress condition is passed, you can take them out again and start translating. If there's a virus infection, you put the viral mRNAs in there, which is good for the cell, but of course is not good for the virus. So how do we get around this? Of course, as you might guess, viruses have evolved ways to get around this, and some of those are shown here. Very common theme in all these viruses. They bind components of stress granules and prevent their formation. Here, hepatitis C virus is actually binding G3BP, which is a component of the stress granule, and using it for its replication complex. We're going to see in a moment that hep C replication and assembly complexes occur around lipid droplets, and the proteins that make up those include G3BP. Poliovirus cleaves G3BP to prevent the formation of stress granules, and other viral proteins bind stress granule proteins. So this is an interesting one. Hunin virus actually makes G3BP part of its replication complex, and the flaviviruses, their structured RNA actually bind these stress granule proteins. All of this prevents the stress granule from forming, and even if they form early in infected cells, the activity of these virus proteins can reverse that. So it's another give and take in a virus-infected cell. Let's turn to metabolism. Viruses impact metabolism as well. Now, you can imagine you're making lots of virus particles in a cell. You need energy to do that. You not only need the biosynthetic capacity of the host cell, RNA synthesis, protein synthesis, but you need nucleotides, you need amino acids, you need fatty acids. You have to be making them often at higher rates. Remember, most of our cells are resting, and then when they get infected, the viruses turn on division, and they turn on processes that are at basal levels. And you need a lot of energy to make all this, 4 ATP, just to make a single peptide bond. And you only get 2 ATP from a single glucose molecule. So that's a lot of sugar that you need to make virus infection. So as you might guess, viruses impact all aspects of metabolism. They turn up metabolic cycles to favor their replication. So let's look at some examples. Here is a very visual example. We have virus replication in cell culture. And these are 6-centimeter plates of cells, and we use a pH indicator in our medium so we know when the pH is neutral. So mock-infected cells on the left, 24, 36, 48, 72 hours, they stay pretty much neutral pH. In the middle, we have cells infected with wild-type adenovirus. You can see by 48 hours, the medium is yellow. It's acidic, and that's because these cells are making lactic acid, which is a product of glycolysis, breaking down glucose. Here on the right is a viral mutant that doesn't replicate very well, and you can see it hasn't turned up metabolism in the cell very much. Those numbers are quantified on the right. We're looking at glucose consumption rate in cells, and you can see the wild type ad in red goes up substantially. This is consumption of glucose, whereas in the mutant virus infected cell, it's low. So that's just one example of viruses kicking up glycolysis. Here is the pathway of glucose metabolism. Glucose, of course, is a major breakdown product of the carbohydrates that you eat. This pathway imports glucose into the cell by a specific transporter. It then goes through the glycolytic pathway. It's converted to pyruvate, and that in the end gives you some energy in the form of ATP and NADH, which can go on and be converted to ATP as well. And a lot of these intermediates can be shunted off, and you can make more ATP from them as well. And finally, two of these products, acetyl-CoA is a precursor for fatty acid synthesis. Fatty acids, of course, are needed to build membranes, which some viruses need, and oxidation of fatty acids will also give you energy, ATP. Up at the top here, this pentose phosphate shunt off of the glycolytic cycle will give you nucleotide synthesis, which of course viruses need to make their genomes. So this can be perturbed in virus-infected cells. Here are two different effects of virus replication on glycolysis. Here we have human cytomegalovirus and herpes simplex virus. So these are two viruses of the same family. They have different effects on glycolysis. So HCMV stimulates uptake of glucose into the cell, stimulates the transporter that brings in glucose, and that gives a lot of acetyl-CoA, which can be used to make membranes. And this virus requires a lot of membrane. On the other hand, herpes simplex virus infection inhibits the uptake of glucose into cells. And you may think, well, that's strange. It's an envelope virus too. The result of blocking glucose uptake is that you have a backup of the pathway at the pentose phosphate step, and that gives you lots of nucleotides. So the idea here is that herpes simplex virus has a faster replication cycle than cytomegalovirus. It needs a lot of nucleotides to be made very quickly, and therefore it has this inhibition of glucose uptake, which gives you more nucleotide synthesis than does human cytomegalovirus. Fatty acids are also important, and their synthesis is tweaked by virus infection. Here, remember, when we metabolize glucose, we end up with pyruvate, and that gets imported into the mitochondria, and there it's made into acetyl-CoA. Acetyl-CoA cannot cross the mitochondrial membrane, so it's converted to citrate, which can be exported, and then converted to precursors of fatty acids back in the cytoplasm. In cells infected with human cytomegalovirus, the action of a number of viral proteins pushes citrate out of the mitochondria into this fatty acid pathway. And if you inhibit in virus-infected cells either fatty acid synthase or acetyl-CoA carboxylase, you can inhibit them specifically with siRNAs. That depresses HCMV replication because they need fatty acid synthesis. Now, you may recognize cyclic behavior of these compounds as a shunt. In fact, not only is acetyl-CoA impermeable to the membrane of the mitochondria, but electrons can't get in either. So the electrons have to be brought in by other compounds, and there are a number of enzymes involved in that. And in fact, VSV stimulates the activity of that shunt in order to make more fatty acids as well. So a really interesting perturbation of fatty acid synthesis. As you might guess, there are human diseases associated with virus perturbation of metabolism, and these are two hepatitis viruses. The liver, as you know, is where you have glycogen produced from glucose for storage and broken down. You can also make glucose de novo. If you're not taking enough in your diet, your body will make glucose. These viruses, hepatitis B virus and hepatitis C virus, which cause hepatitis, infection of the liver, are associated with type 2 diabetes. And so hepatitis B infection stimulates the enzymes involved in glucose synthesis. So presumably this is to provide energy for viral reproduction. But of course, if you make a lot of glucose in the liver, it will spill out into the bloodstream. You have high glucose, and that's one of the signatures of type 2 diabetes. Hepatitis C is also associated with type 2 diabetes, except in this case, infection causes reduced glucose uptake and more synthesis of glucose in the liver. And again, that spills out, gives you high blood glucose, which is not good for you. That's type 2 diabetes, but for the virus replicating in the liver, that's advantageous. The citric acid cycle is also an important metabolic hub in our cells. Of course, it's a big hub of carbon metabolism. It gives you lots of precursors for many compounds. So we input pyruvate from glucose metabolism, and then you can make lots of other compounds. For example, citrate can be used to synthesize fatty acids, and human cytomegalovirus bumps up this pathway, so fatty acids can be made. Vaccinia virus and human cytomegalovirus import glutamine at high levels specifically into the pathway, so it can go around and presumably give rise to more fatty acid as well. And herpes simplex virus, remember I mentioned previously that it needs to make lots of nucleotide precursors, so it particularly upregulates the enzymes involved in going from oxaloacetate to purines and pyrimidines. And these are just examples of this. At every step of this cycle, viruses have impact, so they can bias the cycle to make their reproduction more efficient. Lipid metabolism is also impacted by virus infection. This triacylglycerol at the top left here is a primary energy store. It can be oxidized to provide energy, and of course it's often stored in the form of lipid droplets. Viruses require this for membrane synthesis. Now in us, triacylglycerols are carried in our blood on lipoproteins. These can adhere to the plasma membrane of cells. There's a lipoprotein lipase that will break down this triacylglycerol into free fatty acids, which can then feed into acetyl-CoA, and you can give rise to energy and lipid production, or it can be converted back to triacylglycerols in the cell and stored as lipid droplets. And you'll see in a bit that a lipid droplet is actually the site of assembly for hepatitis C virus. So again, viruses modulate lipid metabolism, they oxidize lipids to provide energy, and they stimulate its synthesis to give a lot of membrane that they require for assembly of new virus particles. Here's an example of the effect of cytomegalovirus infection. So cytomegalovirus requires lipids. It stimulates the production of lipids, but not just any lipid, long-chain fatty acid lipids, which are the main lipid present in the viral membrane. And this virus can stimulate the production of those specifically. The way it happens is the genes for the synthesis of fatty acids and sterols are in the nucleus. They're under transcriptional regulation. And an important regulatory transcription factor is this one, SREBP1M. So when SREBP1M is in the nucleus, you get transcription of genes that will give rise to fatty acid synthesis. Normally, when there's plenty of fatty acids in circulation, the cell senses that, and the transcription factor is kept on the ER. It's inactive because it's not in the nucleus, and it's held in the ER by interaction with two proteins. What human cytomegalovirus infection does, it causes stress in the ER, which causes dimerization of PERK, and that inhibits this yellow protein, which is needed for retention of the transcription factor in the ER. So the transcription protein moves out of the ER through the Golgi, it's processed in the Golgi to the final transcription protein, and then it's imported in the nucleus. And there are also inhibitors to prevent import of this SREBP1 into the nucleus when there are plenty of lipids around. Those are antagonized by a human cytomegalovirus protein. So the end result is increased transcription of genes for lipid synthesis, particularly long-chain lipids, and these are incorporated into the virus envelope. If you inhibit the synthesis of these genes, you inhibit virus production. So they're absolutely essential for virus production. So the virus is upregulating them. Even if you have plenty of lipids around in a cell, the virus will upregulate the synthesis of more lipids for the purpose of building virus particles. Hepatitis C does the same thing. It increases the synthesis and accumulation of fatty acids and lipids. In this case, the target is a transcription factor called FOXO1, which binds to a gene in the nucleus. It gives rise to the synthesis of several proteins, which will in turn increase the level of SREBBP. Remember, that turns on the fatty acid synthesis genes, and you get lipogenesis. Again, in conditions where you have plenty of fatty acids around, FOXO is cytoplasmic, but the virus encodes an inhibitor that keeps it in the nucleus, so it keeps churning out genes that are involved in lipogenesis. Fatty acids not only can be used to build membranes, but they can be oxidized to produce energy. And this virus downregulates genes that are involved in fatty acid oxidation. So FOXA2 is a transcription factor that is needed for the production of proteins that are involved in fatty acid oxidation. The virus encodes a protein that inhibits FOXA2 activity, so no oxidation. It wants all those fatty acids for membrane production. So you decrease fatty acid oxidation, you increase lipogenesis. And here in the upper right is a picture of the effect. These are cells either MOC or Hep C infected. The nucleus is stained blue with DAPI. And then the green color is a dye that binds lipids. So you can see the cells on the left, the MOC infected, are just smattering of lipids in the cytoplasm. And then Hep C infected, the amount of lipids goes way up in the cytoplasm because of these alterations at the level of transcription factors. So it's highly sophisticated, these changes. Our last question is, how might virus infection lead to increased levels of ATP? Stimulation of glucose uptake, increased glycolysis, increased oxidation of fatty acids, increased utilization of glutamine, or all of the above? All of the above, most of you got, that's correct. Glucose uptake, glycolysis, oxidation of fatty acids, and pulling glutamine into the TCA or the citric acid cycle. All right. Now just a couple of examples of one last effect of virus infection on the host cell. This is remodeling. Virus infection remodels many different parts of the cell, including the nucleus, membranes, cytoplasm. But I want to look at two examples of cytoplasmic remodeling. And this one we've seen before. This is co-option of cytoplasmic membranes in the polio-infected cell. I've shown you this lower left electron micrograph showing the cytoplasm of an infected cell. With the little dots are polioviruses that have been made, but the larger double membrane vesicles, these completely replace all the ER and Golgi in the cell cytosol. All those membranes are repurposed to form these double membrane vesicles, and they're sites of virus replication. And that is diagrammed on the right here. So the ER and Golgi is converted to these vesicles that are RNA replication complexes. You can see the RNA being replicated on the surface. As infection progresses, they become autophagic vesicles. These crescents curve and form a double membrane particle. And then later in infection, assembly actually occurs on and in these vesicles. And that can lead to non-lytic release of virus particles, as we discussed last time. And so this requires remodeling of the membranes, and there is an impact on fatty acids as well. So on the upper left is again staining of polio-infected and MOC-infected cells with a dye that binds to lipids. You can see MOC-infected, there's a smattering of dots of lipid in the cell cytoplasm. Polio-infected cell, it's full of fatty acid, and that's because import of fatty acids, not necessarily the synthesis, but import of fatty acids into the cell from the medium is increased by poliovirus infection. That's obviously to provide these vesicles that are needed for replication and assembly. Similar story with hepatitis C virus, which also requires these vesicular compartments in order to replicate. So on the left, early in infection, you see vesicles derived from the endoplasmic reticulum in Hep C infected cells. Some of these are attached to the ER by little stalks. Some of them are single membrane, some of them are double membrane vesicles, but the viral RNA replicates on and in these vesicles. At the top is an electron micrograph showing the vesicles in a real cell. And the next panel is a modeling of the three-dimensional view of that. Later in infection, when virus particles are assembling, they do so also in membranous vesicles, but these are different. You can see the membranous vesicle, which is still attached to the ER here, is now wrapped around a lipid droplet, which is a storage depot in cells of triglycerides, as I showed you earlier. And the viral proteins are actually assembling on the surface of this droplet. Here's the blue one is the capsid protein. And then the RNA will eventually come in here. It's going to be replicated on similar vesicles. And the particle is actually butting into the lumen of one of these vesicles. So it's going to end up forming a complete particle right in the lumen of this vesicle. Lipid droplet is an essential part of that process. And of course, those droplets, as I mentioned before, are produced in part by interfering of lipid metabolism by virus infection. Let me show you one more example just to wrap all of this up and really bring metabolism and remodeling together. This is a pretty recent paper from November of last year, which I think is really amazing. It is studies done with a plant virus, tomato bushy stunt virus, which infects tomato plants, as you might guess. It is a plus-strand RNA virus, icosahedral shell. It also induces membranous vesicles in which RNA replication occurs. And here is the RNA polymerase complex. P92 is the viral RNA polymerase. It's recruited to vesicles. And here is a typical vesicle that RNA synthesis occurs within. One end is open, and the RNA synthesis occurs here. And again, these are similar vesicles to the ones we've already talked about for a hepatitis C virus. But the amazing thing here is that pyruvate kinase binds to the viral RNA-dependent RNA polymerase. So pyruvate kinase is this green protein, PKCDC19. It binds to the RNA polymerase. So it gets pulled into this replication vesicle, and it supplies the ATP that's needed for RNA synthesis. Doesn't that make sense? There's where the RNA synthesis occurs. To ensure that the enzyme has enough, it brings in an enzyme that can generate ATP right in this vesicle. And so what's happening is shown on the right here. There's the polymerase P92 making lots of copies of viral RNA. And the actual step at which ATP is needed, you might think it's for producing the nucleic acid, but it's not. It's actually a helicase that's needed for denaturing the strand, the new strand from the old one as it's made. That's an ATP-dependent reaction. The pyruvate kinase is brought into the replication structure just for that purpose. So that's a beautiful confluence of manipulating metabolism and remodeling the cell. That is the end of our journey through an infected cell in culture. Now we're going to move to bigger stories. That is, what a virus does when it gets into a host. And there is way different. It's not life in a petri dish anymore. Lots of complicating factors. So we'll start that next time.